So good afternoon and good morning for the rest of the people at the other side of the world. My name is Alejandro Caro Quintero. I'm a PhD researcher at AGROSAVIA. This is going to be a very unique presentation because actually we do agricultural research, but because of the pandemic, the government has asked that the research institutes with the labs and the knowledge to support the diagnostic of SARS-CoV2. So in the last 3 months, we kind of shifted our goal in our institution to help some of the regions in Colombia that do not have labs for diagnostics. We are currently have processed around 6,000 samples of different patients. And we at the beginning, we have to shift from our original goal to start helping the country. And with that, we realized that there was a big problem on RNA extraction protocols or RNA extraction kits. Many of the countries at that point were importing kits from the US or from China or Korea, and we were having difficulties to get those kits imported to Colombia and to have the opportunity even to buy them. So we bought some kits at the beginning for the diagnostics, and then we searched around for protocols that we could use in the meantime, so that if we have any shortage of such kits, we will have the possibility of doing diagnostics. So we came across the BOMBIO initiative. It's a multidisciplinary group that has for a long time developed methods for DNA and RNA extraction based on magnetic beads. And they helped us to set up their protocol, and then we have to do some optimization of that. But the main idea is that there is little need of equipment. Most of the RNA extraction protocol, they require centrifuge or vortex or things that our lab has, but probably some others in the remote regions of Colombia don't have. So we decided to go and try this protocol. We have used similar protocols in genomic sequence, amplicon sequence cleaning, and library preparation for Illumina sequencing. So we had already experience on this type of reagents and how to do the cleaning. It was also a very fast protocol. You could do one sample in 35 minutes. It is quite inexpensive, as you can prepare your own magnetic beads, and then it can be fixed for multiplexing if you have the right rags, or if you have a 96 well format, then you could do many diagnoses at the time, do the extractions. So basically the protocol is the one that you see there. You add some lysis buffer, which is a GITC buffer, isopropanol, then the beads and the sample. You mix, you put it on a neodymium magnet that separates the beads from the solution. Those beads have already trapped all the DNA and RNA from the sample. And then you do cleaning with isopropanol and ethanol, and then you resuspend that on water or tea. We started to set up optimize this protocol from BOMBIO, and then we realized that when you have real samples, so we did it with a commercial kit that had a column-based extraction and this magnetic bead kit. And then we realized that in some cases, when the samples were extremely bloody or very dense, we had problems with this magnetic protocol. So then we optimized this, and what we decided after many trials and different tests was to, whenever we have these complicated samples, we diluted our sample 1/5 with sterile saline solution. And then we also add on the step of the lysis 12 microliters of proteinase K and incubate that at 60 Celsius for 10 minutes. And that kind of helped a lot on the extraction of complicated samples. And then we run in parallel qPCR, RT-qPCR detection of samples with samples that we knew were positive and other ones that we knew were negative based on the commercial kit. These are some of the results we have, very fast. This is the CT values for different samples. The different numbers correspond to different patients. And then we have our beats protocol and then the column-based extraction. And as you can see, for the RDRP, we also try with the gene E and gene N. We have very similar values. In some cases, the column-based extractions have lower CT levels. In some cases, our beats have lower CT levels. So it's quite similar, but it's more similar with the RNA-SP marker, the human marker. It seems I have heard that it's possibly because of DNA co-extractions, and then we're amplifying always also the gene, the DNA, and not the RNA. But for the RDRP, the results are very similar. We also wanted to assure that this was sustainable, so we did the quantification of price that will how our protocol will compare to the ones that are the kits, commercial kits that are available, and adding all the reagents and taking into account that importing many of these reagents is super expensive in Colombia. Still very cheap. It's around 50 cents per sample, the RNA extraction, compared to around $10 per sample using the commercial kits that we are getting in Colombia. And this is because of there is a high demand on other countries to do these extractions, and therefore the prices in Colombia have increased 40 to 50% of what they used to be. I think this is all. I just wanted to say that other approximations that the ones that we're gonna see this afternoon, like the LAMP, or even are suitable for doing diagnostics on site in many remote regions in Colombia. There's a lot of people in Colombia interested and trying to do LAMP diagnostics. However, it is important also to have very defined methods for RNA extraction, because that's the main limiting step, I think, in LAMP sensitivity. Is not the LAMP per se, but the quality and concentration of RNA. And I think one of the possibilities that people in other parts of the world could use to diagnostics on site will be to adapt this magnetic bead extraction of RNA that then doesn't require a lot of reactives, and then you don't need centrifuge on site. And with that, I'm finishing my presentation. Thank you.